All right, so this is Floriculture Nutrition, the latest in phosphorus fertility, part 3. We're looking at a late season or a reproductive stage phosphorus deficiency. So again, this is a 3-part webinar, and now we are in part 3, looking at a reproductive stage phosphorus deficiency. So just to go over phosphorus deficiency a little bit, phosphorus deficiency symptoms typically occur when plant tissues comprise less than 0.2% phosphorus. And phosphorus is mobile within plants, of course, and so the source-sink relationships among tissues help to define where the phosphorus deficiency symptoms occur. And typically, because it's mobile, we see the phosphorus moving out of the source tissues or the lower mature tissues, such as the lower leaves, and into the sinks, such as the upper leaves and the flowers. And so this explains why deficiency symptoms typically occur on the lower foliage of your plants. So really quickly, I just want to reiterate what I just said with this diagram and illustrate phosphorus movement within the plant using this mum plant right here. And please keep in mind that this plant is not in flower, and that will come into play later. So typically you provide your phosphorus via the fertilizer, whether it's granular or if it's like a constant liquid feed, you supply it with the irrigation. And so you get the phosphorus in the substrate. The plant takes up phosphorus through the roots, and it moves up the stem and into the upper developing leaves. However, when you cut off that external supply of phosphorus, what you see is the substrate will very quickly run out of phosphorus supplies. And when it runs out of phosphorus, it's going to start having to move those phosphorus resources from the lower leaves up into the upper leaves. And that is what causes the symptoms of olive green leaf spotting, red coloration, etc., that are commonly associated with phosphorus deficiency. So again, the typical symptoms of phosphorus deficiency are often a foliar reddening or purpling, darker green coloration, stunted growth, and delayed flowering. You can see the New Guinean patience on the left there, the left plant grown without phosphorus, very stunted and red or darker green in coloration. And the plant on the right is healthy, green, no signs of phosphorus deficiency. And on the very far right, you see there's a mum plant that's displaying these very typical, common, characteristic symptoms of phosphorus deficiency on the lower leaves, that nice dark purple coloration being displayed there. And also, there are other symptoms. There are the olive green and necrotic leaf spots, as you can see on these two leaves here. The leaf on the left from an ornamental pepper, you can see that it has a little bit of olive green leaf spotting, as well as some of that necrotic speckling or spotting along the leaf margin. And then you can see this poinsettia leaf that also has this chlorotic region within the center of the leaf that has this abundance of olive green leaf spots developing between the veins. So we're talking about this reproductive stage phosphorus deficiency. And these symptoms were confirmed on the upper foliage of a mum crop a couple of years ago. And these symptoms were a chlorosis, necrosis, and olive green leaf spotting on the upper leaves. If you look at the picture on the left, you can see that the leaf has that kind of dull coloration with those very prominent olive green spots. You can see those circled in the red circle there. And then the top shot of the mum on the right, you can see that it's those leaves that are just below the flower buds that are showing these symptoms of chlorosis and necrosis. And so this case caused a lot of issues for a couple of growers, and these symptoms resulted in very significant losses for these growers of around $130,000 in losses due to these phosphorus deficiency symptoms. So you may be asking, why have these symptoms not been reported before? So there's a couple of reasons, and one is that they likely have occurred, but they are just so atypical of what you consider as a phosphorus deficiency that they weren't associated with phosphorus at all. In fact, many times we've heard that these symptoms actually appear more similar to a bacterial leaf spot than a phosphorus deficiency or another nutritional issue. And additionally, with the increased popularity of low phosphorus fertilization, Some growers have probably limited phosphorus too greatly, and that has only occurred over the last couple of years, a couple of decades, as people have moved to try and limit their phosphorus fertility. So our research question here is, what causes these atypical symptoms on the upper foliage? Now, back to diagramming and illustrating how phosphorus moves within the plant. But this time, we're going to look at a mum that is in flower. So what occurs when the plant is in flower, but there's not enough phosphorus available? Well, this time, instead of just having one sink, being the upper leaves, you've got two competing sinks, being the flowers and the upper leaves. And so we determined, we have this bypass hypothesis, where when you run out of phosphorus in the substrate, the roots no longer have any phosphorus to take up. So it starts to move phosphorus out of the lower leaves and up the stem, but it preferentially allocates phosphorus to the flowers and fruit in favor of reproduction rather than its own growth. And it's these phosphorus resources coming out of the source tissues or the lower leaves, bypassing the upper leaves, and that is what is causing these symptoms on the upper foliage. So to test this hypothesis, we did a couple of experiments. We grew ornamental peppers with rates of 0 to 20 parts per million phosphorus, holding everything else constant. And half of the plants were restricted to 0 parts per million phosphorus after 6 weeks. Also, after the pepper experiment, we grew chrysanthemums with rates of 10, 15, or 20 parts per million phosphorus, and half of these plants were restricted to 0 parts per million phosphorus after 4 weeks. And the reasons that we chose 6 and 4 weeks in these cases are because these are the points at which plants began to flower. We suspected that this issue was specifically caused by reproduction, by the flowers coming on. So we cut off the phosphorus at the point that the plants were starting to develop noticeable flowers. So at the end of the study, we divided the plants into separate parts to analyze the tissue phosphorus concentrations throughout the plant. And we did this to illustrate phosphorus movement between the different source and sink tissues. So here you can see Tango Red Peppers, the plants on the top grown with constant phosphorus, and the plants on the bottom grown with an initial rate of phosphorus that was then restricted to 0. And here you can see that really it was only the plants that were grown with 10 and 20 parts per million phosphorus constantly that looked very healthy and were saleable. And with rates of 5 parts per million continuously, that was still too low for this cultivar of pepper. And you can tell because the plant was significantly stunted, and you can see that it had several chlorotic leaves near the bottom of the plant, and in fact, it was just not a very full plant at all. And then when providing rates as low as 2.5 or 0 parts per million, you saw some very, very significant stunting. And however, when you look at the plants that were restricted, you can see that the plant that was grown with 20 parts per million and then switched to 0, although it's starting to look a little sad, it for the most part is relatively healthy when you compare it to the plant that was grown with continuous 20 part per million phosphorus. But when you look at the plant that was grown with 10 parts per million phosphorus and restricted to 0, you can see that this is where the symptoms really came in compared to the constant P plant. And you can see that there were those necrotic leaves falling off. And you can see most of the missing leaves are actually the ones just below the peppers. They're the leaves that were closer to the top of the plant. So that previous picture we showed you was 5 weeks after the phosphorus had been cut off. Here's a picture of the 10 part per million phosphorus plant that was switched to 0 3 weeks after it stopped receiving phosphorus. And you can see that for the most part, the plant was very healthy and green, but some of those leaves just below the peppers have that chlorotic, dull appearance with a little bit of necrotic spotting. So with just 3 weeks without phosphorus, we saw these symptoms starting to come on. And then again, comparing the continuous phosphorus and the restricted phosphorus plants after 5 weeks, you can see that 5 weeks without phosphorus was completely devastating, and the plant lost the majority of its upper foliage. So here I'm just going to illustrate how we divided the plants so that you can see how we went about doing that. we divided the plant first by the peppers, the actual fruit and flowers, the upper foliage and stems, which was about half of the upper vegetative tissues, and then the lower foliage and stems, which represented the lower half of the foliage and stems. And when looking at the tissue phosphorus concentrations in each of these different parts, you can see that in the flowers and fruits of both continuous, the green bars, and restricted phosphorus plants, the black bars, you can see that the flowers and fruit had the highest concentrations of phosphorus. And as you move your way down the plant, you saw that there was a natural limitation in phosphorus. So even plants that were grown with continuous sufficient levels of phosphorus had the lowest levels of phosphorus in the lower foliage and the highest phosphorus levels in the flowers and fruit. And then the upper foliage was somewhere in the middle between those two. But if you look at the restricted bars, you can see that, again, the flowers and fruit had higher phosphorus concentrations, but the upper foliage and lower foliage actually had very similar levels of phosphorus, which is not the trend that you saw in the continuous plants. And so when you compare these concentrations within each different plant part, you can see that the flowers and fruit lost 48% of their phosphorus concentration. The upper foliage lost 68% of their phosphorus concentration, and the lower foliage lost 65%. So again, this illustrates that it was the upper foliage that lost the greatest percentage of phosphorus, and it was the flowers and fruit that lost the least, which indicates that there's this preferential allocation of phosphorus to the flowers and fruit. So moving on to chrysanthemums, here is one cultivar we worked with, Crystal Misty Purple. You can see these plants were grown with 10, 15, and 20 parts per million phosphorus. And what I mainly wanted to show you here is that the plant grown with 10 parts per million phosphorus and then switched to 0 had symptoms of purpling and necrosis both on the lower and upper foliage. Whereas if you look at the plants grown with 15 and 20 parts per million phosphorus, the symptoms were primarily just on those upper leaves. And so here's some more close-up shots of those symptoms. If you look in the foreground, you can see those leaves just below the flowers have a very deep purple coloration. And as you look further down the plant, they have more of that typical healthy green coloration. So it's what you would typically consider a phosphorus deficiency symptom. It's just not in the place you'd typically expect it. And again, here are some more symptoms. We saw a necrotic curling of the leaf margins, and we also saw some olive green leaf spotting within the purple region of the leaf on the top right side of the slide. And here for Crystal Misty Purple, here is the symptom progression. You can see the leaves on the left are green and healthy, going toward deeper red and more necrosis on the right. So you can see that we had a huge variety of symptoms ranging from simply chlorosis to chlorosis and reddening, chlorosis, reddening, and green leaf spots, especially if you look at that lower leaf that is second from the left. You can see that that leaf actually is displaying pretty much every phosphorus deficiency symptom that we've discussed so far. It has chlorotic areas, red areas, necrotic areas, and those olive green leaf spots. So it's important to know that these symptoms can occur either one at a time or all together. So it's important that you recognize and know all of these symptoms. Here's another cultivar that we worked with. This is Little Rock, and you can see it has those upper leaf symptoms of chlorosis and necrosis. However, it didn't display those purple colorations, and that's likely because this plant just doesn't have the same ability to produce the red or purple pigments as Crystal Misty Purple had. And so when you compare the symptoms we see on the left from the study with the symptoms that were recorded by Dr. Whitaker in 2014, you can see that they're very similar with the symptoms of chlorosis and either tip or marginal necrosis on those leaves just below the flowers and flower buds. So just to illustrate how we divided these plants, we did it a little bit differently than the peppers, but pretty much followed the same trend. We divided it first by the flowers, then the upper foliage and stems that represented approximately a third of the upper vegetative tissue, the middle foliage and stems, which represented the middle third of the vegetative tissues, and the lower foliage and stems, which again is the lower third of the vegetative tissues. And when we looked at the tissue phosphorus concentrations, we saw a very similar trend to what we saw in the peppers. First, if you look at the trend in the green bars, you can see that the flowers had the highest phosphorus concentrations, a little over 0.5% phosphorus in those tissues. And as you work your way down to the upper leaves and the middle leaves, you have a significantly lower phosphorus concentration in those tissues, and then the lowest phosphorus concentration in the lower leaves. And again, this is in a plant that was not displaying any phosphorus deficiency symptoms. This is the natural tendency of distribution of phosphorus within the plant. But again, if you look at the black bars this time, you can see that the flowers, although they had less phosphorus than the continuous phosphorus plants, they still had a greater, significantly greater concentration of phosphorus than the upper, middle, or lower leaves. And in fact, all of the foliar tissues had similar levels of phosphorus when compared with the flowers of these plants. And again, when you look at how much these tissues lost by being restricted, you can see that the flowers lost exactly 50% of their phosphorus concentration, but it was the upper leaves that lost out the most. They lost 86% of their phosphorus when compared to plants that were continuously grown with phosphorus. So here we saw that these source-sink interactions led to differences in phosphorus allocation. And if you look at the arrow on the left, you can see the natural tendency in a plant when it's given continuous phosphorus fertilization is that you just naturally have lower concentrations of phosphorus in the lower leaves, and those concentrations get higher and higher as you move up the plant to a point where they are the highest in the reproductive tissues. Now, if you look at the arrow on the right side of the plant, you can see that when you restrict phosphorus, you have equally low levels of phosphorus throughout all of the vegetative tissues and much higher levels of phosphorus in the flowers and fruit. And now, although the levels are similar throughout all the leaves, it's still those upper leaves that ended up losing out on the highest concentration of phosphorus. So again, 86% of that phosphorus was lost, indicating that it is those upper leaves that lose out on the most, and that is why we see the symptoms on those upper leaves. So in conclusion for part 3, we saw that restricting phosphorus upon floral initiation resulted in this upper leaf symptomology that was reported in 2014. Conditions of sufficient phosphorus early on and then restricted later are what led to these symptoms, and the lower initial phosphorus rates led to typical lower leaf symptoms. So it really only occurs when you're giving them sufficient phosphorus, low but sufficient phosphorus early on, and then restricting it later. And so the phosphorus tissue analysis from the peppers and the mums helped to support our hypothesis that phosphorus was bypassing those upper leaves, and phosphorus is preferentially translocating to the developing flowers and fruit. And if you want to know any more information about these studies, please check out our September 2017 edition of Grower Talks magazine. We have an article called Phosphorus Deficiency on Upper Leaves. So please check that out if you're interested. And lastly, I just want to go over some of the overall conclusions from all of my work that we did here. So some of the pros that you can see from using low phosphorus, we saw from these experiments that growers can obtain numerous benefits from low or approximately 10 part per million phosphorus fertilization. And some of these benefits include controlling stretch, enhancing coloration, and limiting your environmental contamination by limiting the excess of phosphorus runoff and phosphorus waste. And unfortunately, there are also some cons that we need to talk about, and that's primarily due to the poor nutrient holding capacity of soilless substrates, such as peat-based substrates. You really have to supply at least some level of phosphorus to avoid deficiency symptoms. As we saw in some of these previous studies, you can go very, very low without running into phosphorus deficiency symptoms, but you really need to apply at least some level of phosphorus to avoid symptoms. And from what we saw in the reproductive stage deficiency studies is that symptom development can occur in as little as 2 to 3 weeks after you restrict phosphorus to 0. So for most species, 5 to 10 parts per million phosphorus will prevent symptom development and will provide you with a healthy plant that has reached its mature maximum size. And so by supplying that recommended rate of 5 to 10 or up in some cases 5 to 15 parts per million phosphorus, you can grow a very healthy plant. And with that, thank you for listening. I'd also briefly like to thank our research supporters that you see listed on the screen. And I would also like to thank all the help from the people in the Floriculture group here at NC State, to Brian Whipker and Ingrid McCall for all of the hours and hours of work that they helped with making all of these studies possible. But then again, thank you for listening, and I'd be very happy to answer any questions that you have. Thanks.